Thank you for joining this installment of Boyce Thompson Institute's Breaking Ground discussion series. My name is AJ Bushy and I'm in the communications department here at BTI. And for this month's Breaking Ground, we welcome Dr. David Stern, a plant scientist and BTI's president, who focuses on the molecular mechanisms behind photosynthesis, which is the process that plants use to convert light energy into chemical energy in the form of carbohydrates. And today we'll be talking about David's research into different enzymes along the photosynthetic pathway and how scientists might be able to tweak them to boost agriculture outputs. Hey, David, how you doing today?
Hello, everybody. Good to see you, quote unquote, see you, and see some old friends there in the participant group. Great to have you join us.
Yeah, this is going to be a good one. Thank you all for joining us. And before we get going, a couple quick housekeeping announcements, which will be familiar to those of you who have joined us before. During this session, which will be about a half an hour, all participants will be muted, and you can ask questions in the chat, and then we'll have a Q&A session after that conversation, which will also be about a half an hour. So we'll have an hour total, have about a half hour conversation, and then a half hour Q&A. And I've also enabled a live transcript button, so you should be seeing closed captions, and if you would like to turn that off, you can do it with a live transcript button down at the bottom of your screen. And thanks again, everyone, for joining us today. Thank you, David. And I guess my first question for you, David, is if you could just introduce yourself and tell everyone how you got into plant science and how you ended up at BTI doing what you're doing.
Sure. Well, I was privileged to come from a science family. My father was a botanist, and so I saw what a lab was like. I spent a little time there goofing off when I was a kid. But when I went to college, I first was pre-med, and then I kind of got disillusioned with that work. Didn't seem like the right thing for me, but I did take a molecular biology class that I absolutely loved. This was at the dawn of molecular biology, not too long after DNA had been figured out. And I asked the professor if I could work in his lab, but he worked with frogs. That was animal biology. And the first time I had to basically dissect a frog, I became in my heart a plant scientist. And when I finished undergraduate, I did a master's degree at Cambridge, and that was a plant science master's in one of the labs that was, I suppose, somewhat of a pioneering lab in plant science. And I got lucky, which is any part of being good in science is getting lucky. I got real lucky that first year, and it got me real excited about being involved in understanding how plants had evolved and how they function. And it kind of went from there in a pretty standard academic path. And when I started looking for a job where I could scratch that research itch, BTI came along kind of near the end of my job search. And I thought, wow, a research institute, a place where I can go and really focus on discoveries and maybe not so much on teaching large courses and things I didn't think I was very good at. So I came to BTI in 1989. That's a really long time ago, AJ. And
Yeah.
BTI has been my life then for the last 32 or so years.
Wow.
Been through a lot, but the thread of photosynthesis really runs through my whole career here. Maybe not unusual for scientists. When you fall in love with something, you don't let go. I guess the slide shows on the left, my current lab team. The littlest one isn't part of the team yet, but the others are. They are a wonderful group of people who give me a lot of freedom to do my job as president, plus help the discovery research along. And then I have hobbies on the right. That's my son Ben there beside the tractor. So we do live on a farm, and I sell down at the farmer's market, and living the dream. So that's how I got here. And I am going to step down as BTI president sometime in the next 3 years. And I'll stay here and do some research for a little bit after that. So that'll be an interesting change in life as well.
And I put a little link in the chat if anyone would like to donate in David's honor as he steps down from president. But I'm glad you'll still be around. You'll still be doing some of your research.
Hold that thought. Yeah.
What's that?
Hold that thought.
So you mentioned photosynthesis. So if you could kind of go into some details there, I think most of us learn that photosynthesis is how plants take sunlight and carbon dioxide, right? They breathe in carbon dioxide, they breathe out oxygen, and they somehow magically make sugar. So what's really going on? What are you looking
Yeah,
at?
I think it's pretty amazing, really, that light could shine on a plant and somehow that energy from the light could be captured and, as you say, AJ, basically turned into sugar. The light itself doesn't get turned into sugar, but the energy from the light gets stored by the plant through use of chlorophyll. So that's the green pigment that you see in plants. And it's able to harvest that light energy and store it in the form of a little chemical messenger. And that's energy. And that energy is like a little battery that's used to allow carbon dioxide, which comes in through small holes in the leaves called stomata, to enter that leaf and be incorporated into larger molecules that eventually turn into sugars. And in the process of harvesting that light energy, water gets split. So we water plants, the water gets split, and the water, if you remember your early chemistry, water is H2O. So the O from that is oxygen, and the oxygen is released from the plant. So the hydrogen is used as part of that energy process. So really what happens is plants power the whole planet through sunlight. They give us the oxygen we breathe. Without them, of course, we'd have an even worse CO2 problem in the climate. And so all animals ultimately depend on photosynthesis to live. So really, sunlight powers the whole planet. Now on the simpler diagram on the left is what I just explained. On the right says, well, the chemistry gets more involved than that, but you can still see those little green discs. Those are the light harvesting pigments, the chlorophylls. The sunlight impinges on those. And again, those pigments are designed by nature to capture light energy. You see that water being split, oxygen coming out the bottom, and then those little chemical messengers, those things like NADP, ADP, phosphate, those are being used to incorporate carbon dioxide into this pathway. It's a biochemical series of chemical reactions called the Calvin cycle, sometimes called the dark reactions. And then out the bottom, why you get sugar. So the plant harvests that light, turns that energy into chemical messengers, and uses that energy to pull carbon dioxide into a biochemical cycle that ends up delivering sugar, which is then transported throughout the plant and used to make things that you like to eat. For example, the sugar in your tomatoes or your peaches or your plums and so on and so forth. So the building blocks of life all start with photosynthesis.
Yeah, and those and some plants kind of split those light reactions from the dark reactions up, right? That's
Well, we call them the light reactions, the light independent reactions. So the
okay.
Calvin cycle doesn't need light to function, but it does need those chemical messengers, and those chemical messengers do depend on light. So you can't do photosynthesis without light. When it's dark, what your plants are doing is they're taking stored starch and converting that into sugar and sending it out to the plants. So plants do grow at night, but they don't grow using photosynthesis.
Gotcha. That makes sense. And this also ties in with different types of photosynthesis, right? You're looking at C4 plants.
Yep. So this C3 versus C4, it's not a contest, but there are two major different kinds of photosynthesis. And basically behind this is the fact that, as we all know, plants and animals, but plants adapt themselves to environments that they live in. So the dominant form of photosynthesis on Earth, if we just think about land plants here for a moment, is C3 photosynthesis. Maybe 97% of plant species do perform C3 photosynthesis, but the other 3% perform C4, and they're quite different. C3 is kind of where everything that happens is in the same place. And in C4 photosynthesis, the reason that you see on the right, you see two boxes, is because the process is split into two parts that are in two different kinds of cells. Now, why would plants do this? Well, C4 photosynthesis has evolved dozens of times independently. Even though it's really different from C3, plants have figured out how to turn their prior C3 photosynthesis into C4 multiple times in evolution, especially when plants live in stressful environments like the desert, in the tropics. Most C4 plants have evolved in tropical or arid environments. And the reason is that the plants are more economical with their use of, with their photosynthesis. And because photosynthesis involves incorporating CO2 from the atmosphere, plants have to open up their leaf openings, these stomata, in order to let air in. And if you're in a desert and you open up your leaves, you lose water from a plant, right? So plants want to conserve water in hot environments. And the more efficient they are with photosynthesis, the more time they can spend not losing water, transpiring. So C4 has evolved under that context. And in these diagrams, there's two little, I guess, kind of daisy chains of Cs that are stitched together, and they have ovals around them. And the one that's on the left under C3, you see that's a 3-carbon molecule. Each of those Cs represents a carbon. And that's one of the main participants in C3 photosynthesis. So we call it C3. It's got 3 carbons. In C4 photosynthesis, you see that chain of 4 Cs. And that is the form of carbon that goes between the mesophyll cells, which are shown at the top, and the bundle sheath cells, which are shown at the bottom. So it's a C4 or a carbon, 4-carbon chain that shuttles between cells, and that's why it's called C4 photosynthesis. It's not always the same chain of 4 carbons, but it's always 4 carbons, and that's why it's called C4. And what really happens in C4 that's different than C3 is that carbon dioxide, or carbon, is fixed or assimilated twice. So in the C4 plants, in the mesophyll cells, which are at the top right, CO2 comes in and it's actually absorbed into that C4 compound. That C4 compound goes from that cell type on the top, which is nearer to the edge of the leaf, to the bundle sheath cells, which are shown at the bottom, which are more in the interior of the leaf. And that CO2 is released again. And then it's fixed by this enzyme, the one that my lab loves, called Rubisco. And CO2 then in C4 plants is fixed twice. And what happens is when it's re-released in these bottom cells, it's very highly concentrated compared to in the atmosphere. And that makes the enzyme Rubisco much more efficient, because Rubisco is a very inefficient enzyme, and it can be driven to more efficiency by higher amounts of CO2. So in the C3 photosynthesis, the amount of CO2 is simply what's in the atmosphere. In the C4 plants, it becomes highly concentrated through this double mechanism, making the Rubisco enzyme much more efficient. But it takes a lot of energy to make these complicated cells. You got to make 2 different kinds of cells. You've got to put enzymes in one cell versus the other cell. You've got to move things around, these carbon chains. And so C4 photosynthesis has evolved where it gives an advantage to the plant. But because it's only about 3% of plant species, it's less studied than C3 plants. And that's noted there on the left side of the slide. What did I leave out, AJ?
I don't think you left much out, but I was just gonna add that one of the reasons I always thought Rubisco was pretty interesting is that it can use oxygen or carbon dioxide to do a reaction, right? So that's kind of the competition is if you're just using in the atmosphere like C3 plants do, then there's a lot of competition there.
Right. So thankfully, there's a lot of oxygen in the atmosphere. We'd all be in trouble. And it's a lot more than there is of carbon dioxide by percentage, like I said, or we'd be in big trouble. So when the atmosphere enters a mesophyll cell in a C3 plant, oxygen can bind to the Rubisco enzyme and compete with carbon dioxide. And so Rubisco will react with either one of them. If it reacts with oxygen, it produces essentially a waste product that has to be recycled. And so that's called oxygenation instead of carboxylation. Now, because the CO2 concentration in C4 bundle sheath cells is so much higher, there's very little, if any, competition from oxygen. So that's good for the Rubisco enzyme. And so, yeah, we're going to get to this later, but if you did try to make Rubisco smarter and tell it not to bind oxygen, turns out that it gets worse at binding CO2 as well. So it's very hard to make the so-called discrimination of the enzyme better. Nature's solved the problem as well as it can, it appears, for now.
Well, O2 and CO2 are pretty close together, at least on paper.
On paper, yeah. Well, you'd have to be an enzyme to understand what the pocket looks like where these things bind, but the competition is significant. Again, because oxygen's maybe 20 times more abundant than CO2 in the atmosphere.
So so when you're working on boosting photosynthesis, which is in the title, are you working on Rubisco? I mean, what are the limiting steps there? How can you do that?
Well, Rubisco is an interesting thing. Everybody should know that name because it's the most abundant protein on the planet. There's nothing more abundant in the planet than Rubisco. And so it's been studied quite a lot. Again, for example, to try to make it more efficient or you know, whatever, or faster. So it's a pretty obvious target because it's the gateway to incorporating carbon dioxide into sugars in the plant. It can't grow without it. It's abundant, it's essential. Now we got interested in Rubisco for reasons that are typical of science. We didn't mean to, but we got into it. But now we got into it and we're fascinated by it, right? So the form of Rubisco that you might find in a plant like a corn plant, the ones we're primarily talking about today, is the one that's shown at the bottom, and it's called L8S8 because it has 8 so-called large subunits. Those are the green ones there that are they're pairs of 2. It's 4 pairs of 2, and then there's 8 of these small subunits, this S8. And those are those little blue cylinders. There's 4 on the top and 4 on the bottom. So that's the enzyme we call Rubisco. It's so-called hexadecamer because it has 16 pieces. And the challenge for the plant really is to put those 16 pieces together properly. So first of all, the plant needs to make roughly equal amounts of the large and small subunits. It has to fold those proteins up once they're made. That's the top part of the diagram. And then there are these so-called helper proteins or chaperone proteins that help put that protein together, that Rubisco. So first we put 2 L's together to make 2 L's, L dimers, if you want to call them that. Then we assemble those dimers, and then we add the small subunits. And that process is pretty intricate. It's now been done by another group, a group in Germany in the test tube, and they've shown how intricate the process is. And it involves those 4 molecules that are in the square in the middle, so-called RAF1, RAF2, BSD2, RBCX. And my lab, in collaboration with another group in Oregon, discovered these RAF proteins, RAF1 and RAF2. And RAF1 in particular, we got interested in because we thought maybe RAF1 is somehow, the amount of RAF1 is a limitation to the formation of Rubisco. And if maybe we could provide more RAF1 than a plant would normally have, and more of the constituent subunits, these L and S's, maybe we could make more Rubisco. And to get back to the previous slide, we focused on corn as a C4 plant. And the reason is that C3 plants make a lot of Rubisco already. That's why it's the most abundant protein in the world, because most plants are C3 plants, and C3 plants make lots of Rubisco. So when you make a lot of something, it takes a lot of resources. In the case of plants, nitrogen. Whereas in C4 plants, they have much less Rubisco. So there's more potential to make more without affecting the nitrogen budget of the plant. So that was our strategy. Make more of the constituents of Rubisco and increase the ability of plants to assemble it, do that in a C4 plant, and see what happens. That was our approach to the problem initially. We had no idea.
Happened?
What happened? Yeah. So I think Mike should show the next slide. And so what happened is shown on the right, really. So these are 4 maize plants. These pictures were just taken a couple of weeks ago by one of my students. And what you see on the left is what we call a WT, or a wild type plant. So this is just a maize plant. It's just grown in a pot. Now, if we give that plant an ability to make more of this helper protein, this RAF1, the plant doesn't look much different. When we give the plant the ability to make more subunits of Rubisco, the L and the S, that plant's called LSSS. You see that plant looks bigger. And when we give them that helper protein and the subunits, that gives us the biggest plant of all. And size is only one measure, of course, of how you've changed the plant, but these so-called RAF1 LSSS plants, we call them our turbocharged maize because it sounds cool. These plants, they grow faster, they flower sooner, which farmers care about. They flower a couple of days sooner. And as far as we can tell, they make more grain, although we really have to try that out in the real world. Right now, we can only grow these in the greenhouse because they're genetically modified plants. So they're interesting, they're fascinating, they're consistently faster growers, and they're more resilient. And what's shown on the left here is what happens if you plant corn early and a frost hits. The corn plants are very unhappy. They'll even die. So this sort of bleaching of the plants is typically happens when farmers plant their corn and they get a late frost or a late chilling, almost a freeze in, say, May. We were wondering if our turbocharged plants would be more resilient to chilling, because when maize plants get chilled, the amount of Rubisco goes down. They can't keep their Rubisco around. And so these plants, these RAF1 LSSS plants, are preloaded with extra Rubisco. And we were able to show that those plants were much more resilient to chilling stress. So we can make the plants grow faster. We can make them more tolerant of chilling. And these seem to be two really interesting traits. And my student, Coralie Celeste Smith, you see her last name there. She was a wonderful graduate student who published the two papers. She was the leading author on these papers that described these the plants and their properties.
And so what other, are you looking at any other targets in addition to Rubisco?
Well, this gets to the question of what does it mean to optimize photosynthesis, really? So in our research, we started with Rubisco. As I said before, it's a very obvious target. And we got a boost. We got bigger plants, we got a little more grain, we got a little flowering early, and that was really cool. But the question is, now what is limiting these plants from improving their performance? And in this particular slide, you see these is a complicated pathway. This is how the Rubisco enzymes put together. Again, on the right, you see those smaller subunits. They're in magenta, and then the white is the large subunits. But these are now real molecular structures. Anyway, the three molecules that are circled in blue are the ones that were used to produce those turbocharged plants. But the interesting thing about Rubisco, or one of the interesting things about it, is that when Rubisco is made, those 16 bits are assembled together. The enzyme doesn't work. It's inactive. And you see that oval, or that rounded rectangle saying activation. So on the left side of that, in that rectangle, you see Rubisco is there. It looks right, but it doesn't work. And it has to work. In order to work, it has to use this other enzyme called RCA, which is called Rubisco activase. And RCA removes molecules from the interior of Rubisco that are there when it's made, that keep it from doing its good work. It's like if you got a credit card that you hadn't activated it yet, it doesn't work. So RCA is responsible for turning Rubisco from an inactive enzyme to an active enzyme. And what we found in our high Rubisco plants is that a lot of the extra Rubisco they were making simply wasn't activated. So our simple idea was just to make more activase, make more RCA, and there, presto, we would increase the amount of improvement from that extra Rubisco that we knew was present but not active. So that's one of the targets. The other target is this other enzyme shown on the right called PBDK. PBDK is not in the same kind of cell as Rubisco. It's in this cell where CO2, carbon dioxide, is initially assimilated. And because the C4 photosynthesis is a cycle, the CO2 is fixed in one cell, it's incorporated into a C4 compound that goes into the other cell. And then the remainder, once Rubisco does its work, comes back and it has to be recharged. And PBDK is an enzyme that recharges it. So our idea is that PBDK needs to be increased in order to better balance the cycle. In other words, if your cycle is out of sync, if it's going faster in the bundle sheath cells than it's going in the mesophyll cells, ultimately the plant will run out of something. And we think it may run out of a PEP, which is something that is made by PBDK. So those are two targets that we're working on. I couldn't tell you yet. We might be lucky, we might not be lucky, but that's science. That's why BTI is here, is to support research. We'll see where it goes.
So this kind of brings up an interesting thing we were talking about earlier about trade-offs, and how there are so many different moving pieces, so many different targets, so many different molecules involved in photosynthesis. If you boost one, it might monkey with another one, and how do you find the optimal, how do you optimize that? How do you figure out exactly what will work and in what combination?
There's very few good things about COVID, but one of them is that everybody had to figure out how to re-optimize their life. When things shut down, when schools shut down, you can't get daycare if you're a parent, for example. There's so many things that we had to rejigger. Like you couldn't just go remote unless you had a home office. You couldn't have a home office if you didn't have internet, you know. So we all went through this process of understanding, let's go, the supply chain, toilet paper, like all of these things we've learned and remember from the pandemic point to how interconnected, you know, all the bits and pieces of our world is. And photosynthesis is really no different. So we might be focusing on that reaction that takes CO2 and puts it into sugars. But what about water? What about nitrogen? What about the efficiency of light capture? What about dealing with temperature extremes in terms of water loss, and so on and so forth? So if one zooms out from our own work, you'll see that we're working in sort of a tiny corner of photosynthesis. There are people who are interested in helping plants waste less light. That's probably quite important. But then you'll have more energy to do the next piece, which is incorporating CO2. There's some people focused on how fast CO2 moves through cells, from one cell to another, and so on and so forth. So optimization of photosynthesis, you know, if you read about it, is many chapters long. There are many, many ideas. And so when I started in biology, you know, 100 years ago, all we did was sort of guess. We would do something and see what would happen. It's kind of simple-minded. Now we're getting into an era where we have big data. We have computer models. We have artificial intelligence, although the IQ of that intelligence is still pretty low in many cases. So scientists now are trying to predict what experiment will and won't work. And I say in photosynthesis, we're able to predict within smaller pieces of the puzzle what might happen if we change something. If we change this bit of Rubisco, what it might act like. But putting that into a context of photosynthesis more globally, how will this corn plant really grow if we put it out in the field? We don't know how to do that yet. That computational ability is still far off in the future because plants are just too complicated. We aren't smart enough to teach our computers how to figure it out for us. So there's a mix now of relying on computational predictions and simply trying something in the lab. I'm a little old school. I think we're not smart enough to know the result until we do the experiment. So that's what we do. But in the future, it'll probably just be the robots teaching the other robots. And I don't know what people are going to do with their time anymore. You know?
Yeah, those will be the days, right? So what impact do you see your research having on agriculture?
Yeah, so this slide is quite interesting because it shows the yield. This is corn, not yield over years. In years, corn's been growing for a long time in the US. Took off in the Green Revolution, and it's continued to increase. So seed companies have found ways to increase yield, but it's getting harder to increase yield. But the idea of increasing yield is a couple-fold. It sounds good, first of all. It seems like getting more out of a plant would be better if you're growing it for a crop. A bigger head of broccoli seems like more broccoli for your money, right? So it also impacts, so it's better for a farmer. So if a farmer can produce 180 bushels per acre versus 140, that's money in the bank if the price per bushel doesn't go down, which it often does. So, you know, milk is another thing. You make too much of it, price goes down. So yield is good though, always in terms of land use. So if we needed in this country to produce a certain amount of corn and we could do it on less land, wouldn't that be great? Because we could use the land for something else. Let's plant some trees and sequester some carbon, or let's grow maybe some vegetables that are needed for some newfangled meat substitute beyond burgers or something like that. So I think, you know, if we increase the efficiency of growing, again, if we use this more resources, more water, more fertilizer, we really haven't gained a whole lot. But if we can grow the plants with a higher yield because we're using sunlight more efficiently, I think we all stand to gain. So it's not so much that we need more and more corn, although there are people who think we do. It's that we need to use less resources to get more out of our agriculture. It's good for the farmers, it's good for the environment. So that's part of the motivation. I think another motivation for this kind of research is climate resilience. So it's hard to think about chilling stress when we think, well, the planet is warming, but at the same time, there's more volatility in the climate. So if spring comes earlier, farmers plant earlier, but then they're more prone to these early chilling spells. Plus, when corn matures later in the summer, if it can mature before the worst of the summer heat and drought comes, then again, that saves yield and saves a lot of resources. So whatever kind of modification to plants we use, whether it's a kind of a GMO approach, whether it's a plant breeding approach, gene editing approach, they're all designed, I think, should be designed for resilience, number one, and number two, for land use considerations.
Okay, if folks want to start asking some questions in the chat box, we can start our Q&A. But I'll kick one off. Since you're working on C4 plants, is there any potential for your findings to be used in C3 plants, since those are 97% of plants, or the number that you said?
I would say I don't know yet, but I will say that there have been huge investments, a lot of them by the Gates Foundation, but others as well, to turn C3 plants into C4 plants. So rice has been a very popular target for a couple of decades now, because, as I said earlier, C3 photosynthesis uses a lot of nitrogen, and there's a lot of wasteful oxygenation. And so much of what we've learned about C4 photosynthesis has actually been gained by trying to turn a C3 plant into a C4 plant, which is kind of curious. Nobody's trying to turn C4 plants into C3 plants that I know about. So I guess all knowledge is good, but how our particular approaches could be extended to C3 plants isn't clear to me yet.
Okay, we have one question in the chat box. How does corn C4 plants use less nitrogen for Rubisco? Is it because fewer cells, bundle sheath cells need Rubisco?
Yeah,
good question.
Thanks for the question. Yeah, so when Rubisco is more efficient, one simply needs less of it. And so, yes, there are fewer bundle sheath cells than mesophyll cells by far. But in addition, C4 plants just don't need as much Rubisco. So if you ground up a typical C3 plant, like a spinach leaf or something, and looked at all the protein, you'd see this big fat Rubisco band. You wouldn't see it in corn. So corn, about 5 to 8% of the nitrogen goes to Rubisco, and it might be upwards of 30 or 40% in plants like spinach. So the nitrogen budget is dramatically different in those two types of plants. And it's driven by the efficiency of the enzyme, and also, yes, the localization, but I think mostly by the efficiency, which is dependent on the localization.
So if there are fewer bundle sheath cells than mesophyll cells, is it like 10 mesophyll cells all sending their carbon dioxide to a single bundle sheath cell, or how does that work? Is there like a direct pathway? Are they all linked to a single?
That's a great question. Yeah. So I don't really have a great answer to that question, but I mentioned diffusion earlier. So the movement of metabolites. So a mesophyll cell that is far away from a bundle sheath cell or farther away, that C4 compound has farther to go. So diffusion is an issue, but yes, they're outnumbered, absolutely. And I haven't really thought about that, but there is definitely a numbers game. So it's likely that there's less CO2 fixation per mesophyll cell because it would be out of balance. So yes, but they do other things. Mesophyll cells aren't simply there to fix carbon. They do a lot of other things as well. But yeah, there's a big difference in numbers. So basically, if you think about a corn plant, the leaves are kind of striped, and those little stripes are the veins of the plant. So, and surrounding those veins is what's called the bundle sheath, because they're called bundles, vascular bundles. Those are the conduits, if you will, the pipes that take sugars and waters through the plant. And right surrounding them is the bundle sheath. So when the bundle sheath fixes carbon, it turns it into sugar. They're transported right into those pipes, the vasculature, to distribute the sugars throughout the plant. And the mesophyll is outside of that. You don't have that kind of structure in a C3 plant typically. The veins are all over the place. If you look at your house plants, you can tell the C3 plants, there's kind of spidery veins going everywhere. But in C4 plants like maize, there's stripes. It's like a highway, and the bundle sheath surrounds the highway.
Makes sense. Do you have an explanation as to why your boosted maize is more chilling tolerant, given that it's Rubisco that is increased?
Right. So there's a couple of kinds of C4 plants that are, it's one, there's another plant called miscanthus. It's, I guess it's been thought of as a biofuels plant. It's just a huge plant. It grows enormous. And it's actually tolerant of chilling. So what's the difference between maize and miscanthus? Why is maize and miscanthus chilling sensitive and miscanthus chilling tolerant? And one of the observation was that in miscanthus, when you chill the plants, Rubisco amount of Rubisco stays the same. The protein doesn't disappear, but in maize it does. So the amount of Rubisco in maize, it somehow the protein becomes unstable, or the plant gets rid of it on purpose when chilling occurs. So in these turbocharged plants, they're preloaded with extra Rubisco. So our plants do lose Rubisco upon chilling, but they started with more. So they do better than the plants that haven't been modified because they have a bigger reserve of Rubisco when they enter that chilling cycle than a normal plant would.
That's interesting. Let's see. It appears there's no difference in corn plant growth between the RAF1 or LSSS overexpressed lines. Does this mean RAF1 is not limiting and overexpression of the LSSS is enough? I think the pictures did look pretty close.
Yes, right. Of course, that's one picture. Yeah, this is, we've pounded our heads against the wall on this one for years, because these LSS only, SS only plants, they do get bigger, but they don't really have boosted photosynthesis. So we do not know why they have better growth. But when we look at photosynthesis itself, for example, the rate of carbon assimilation, the LSSS plants aren't any better than the control plants or the RAF1 plants, but they are growing faster. And we do not know why. We have theories, but we have no answers. And every time we want to get money to answer that question, people say, focus on the plants that you already know about. So that's what we've done.
Is there any impact on the nutritional distribution within a corn kernel when increasing yield through genetic manipulation? That's a good question.
Yeah, that is a great question. I mean, you know, we sometimes think of corn as something that you pop, or something that is sort of, you know, turned into ethanol or ground up, and I don't know. But there's many, many different components of the corn kernel that are normally separated out for all sorts of industrial processes. There's a book I've read that's basically it's called, you know, We Are the Corn People. Like, you're eating corn all the time. No, you may not know it. You're using it for all sorts of things. It's amazingly versatile. So there isn't one kind of corn. You know, on our farm, we grow some corn mainly to feed the chickens, but other people grow corn to harvest the biomass, the leaves, for silage. There's all sorts of different uses. So there is genetic engineering to change the starch content of corn, to change the oil content of corn, for example. In our own case, what we are trying to do is to get these plants, as I said, into the real world. And we have some technical roadblocks to doing it. We're trying, and we're working with a group headed by Steve Long at the University of Illinois to get the right genetics into these corn plants so that we can make meaningful observations in the field. And at that point, we'll be able to do an analysis of the grain. But right now, we don't have any meaningful way to examine it.
I think they can only be in the greenhouse. Is that?
Yeah, I mean, we know that the grain is, we count 100 grains and then we weigh them. We know they weigh a little more from the turbocharged plants, but that's all we know. We don't know what weighs more. Was it just bigger and everything is proportionally the same or not? We just don't know.
Right. Is that dry weight?
Dry weight, yep.
Right. So you know it's not water. That's something.
Fair enough.
Oh, I asked that one. So what about high temperature? Are these plants more resistant to high temperatures?
Right, that's a great question, because stresses come in packages, right? If you get drought, you usually have heat, for example, not always. So we've published data on heat. The heat data was published with the chilling data because we looked at heat and chilling, and then we looked at drought separately. And what we found is that these plants don't have any particular increased tolerance of heat or drought, nor do they have decreased tolerance. But when the temperatures go back to normal, or the water goes back to normal, they outgrow just like they did before the stress. So they're more resilient in that sense that they don't lose their ability to grow faster just because they've been through a heat stress or a drought stress. But during that stress, there was no advantage at all. No disadvantage, no advantage.
And you said they flower earlier, right? So
A couple of days, yeah.
Yeah.
But again, we grow corn on the farm, and they'll ask you at the store, do you want 93-day corn or 95-day corn? I mean, 2 days matters in the farming world. You may not believe it, but it matters. Or when you plant your home garden, you look at your beans, some of them will say, I don't know, 57 days, some of them will say 59 days. So it's a pretty important parameter in the world of seed and agriculture.
Mm-hmm. Do we know at an evolutionary level if RCA, the activase, or any other unknown regulation downregulates Rubisco in mesophyll cells?
Okay, so in C4 plants, there isn't any Rubisco in the mesophyll cells. You got to be clear about that.
Maybe bundle sheath cells.
Maybe bundle sheath cells, right?
Yeah,
so is Rubisco regulated? Absolutely. So in the absence of activase, of course, all the Rubisco is essentially inactive. And there are these inhibitors. They're sometimes called misfire products. Rubisco accidentally makes things that it didn't mean to make, and those go back and inhibit the enzyme. So they're kind of a key that fits in the lock but doesn't open the door, if you will. And so there are labs that work on these inhibitors and these inhibition processes. And one lab had the brilliant idea of taking the plant's ability to make the inhibitors away by getting rid of another enzyme, for example, this so-called, we call it an unknown regulator. They actually knew what it was. Well, when they took away the plant's ability to make the inhibitor, it did make less of the inhibitor. But then Rubisco was unstable. The protein went away. So there's this theory out there that the inhibitors actually help to stabilize Rubisco. So maybe the plant is calibrating how much Rubisco do I need to be active right now? And any other Rubisco that I don't really need, I don't have anything to feed it. I don't need the activity. I'll just stabilize it. I'll put something in it, this inhibitor that keeps it around, but it doesn't get in the way. It doesn't suck up energy. It doesn't suck up, substrates like these chemicals that it would use to make sugars. So again, it points to the trade-offs when you try to engineer. You think, well, having all the Rubisco active, what's the problem? Well, apparently there's a problem because plants shut it down when they don't need to use it. So it's very curious, but it's a great question.
So it keeps it around if it might need it later on.
Well, it can activate it, in the blink of an eye, rather than saying, well, I have to make all of these 16 subunits and stitch them together and activate it. It's immediately available.
That's interesting. Does PEP carboxylase have any role in bundle cells?
Bundle sheath cells? PEP carboxylase does not. It's only found in the mesophyll. PPDK, on the other hand, is found in both cell types, mostly in the mesophyll, but also in the bundle sheath. We don't know what it does in bundle sheath cells. There's theories about it. It does point to the fact that a lot of the constituents of photosynthesis, some of these small molecules that are part of the carbon shuttle between the mesophyll and bundle sheath, or things that are part of the so-called dark cycle or carbon cycle, these are very common chemicals that the plant uses for many, many things. So again, one cannot even look at the complexity of photosynthesis in isolation, because the plant is using some of those chemical components for other things. So individuals who try to tweak photosynthesis sometimes find out that they accidentally affected another process that they weren't trying to tweak. And one nice thing about Rubisco is that it's only involved in photosynthesis, and Pepsi, as far as I know, is the same. It's only involved in photosynthesis, but it's not thought to be a limitation that is worth trying to engineer. Although there are people, again, who are trying to increase the amount of Pepsi to see if that, improves photosynthesis.
There's one more question. Kind of talked about it. How does temperature affect Rubisco? We've kind of talked about the engineered plants, but is there anything else to add about how temperature affects Rubisco?
Yes, there is. So every enzyme, has an optimal temperature. Nature's designed them that way. So if you went to Yellowstone Park and went to a thermal vent and got an organism, a photosynthetic organism out of it, probably its Rubisco would be adapted to high temperature functioning. And if you put it at the temperature the corn grows at, it wouldn't do very well, and vice versa. So nature optimizes enzymes for the environment that the organism typically lives in. Now, also, almost all enzymes are worse when it gets colder. They slow down. Everything slows down. Things move slower. So, we all move slower when it's cold, or we move fast to get in somewhere where it's warm, right? So, that's normal for things to go slower when they're colder. But where Rubisco has a bigger problem is when it gets hot. And the reason is not because Rubisco itself is so heat sensitive. It ultimately is sensitive to heat like everything, but Rubisco activase is extremely heat sensitive. So activase basically dies at a temperature of about 95 to 100 degrees. And most like corn activase becomes almost useless. And so that means that the Rubisco activation state, the amount of that Rubisco protein that's capable of doing photosynthesis, plummets in heat. So on the other hand, there are plants that have Rubisco activase that does very well at high temperatures. And as you might expect, you find them in the desert. So one of our projects is to take activase from agave, which is a desert plant, and put that into corn and see if our corn now thrives at higher temperatures more so than a normal corn plant.
There's no results there yet though, right, David?
Well, the plants are in the greenhouse. Yeah.
I don't know if we should talk too much.
No, we don't know yet. We really don't. If I knew, I would tell you. Actually, it probably have to kill you if I told you, but no, we don't know yet. We really don't.
That'd be hard over Zoom.
Yeah.
Well, that's the last question, but there's one other thing. If anybody else has any more questions, please put them in the chat. There's one other neat thing about Rubisco that I've always thought was cool since I learned about it, which is that, we talked about the long strand and the short strand. One of them is encoded in the chloroplast DNA, and the other is in the plant's nuclear genome, which I always thought that was fascinating, that somehow, two completely different genomes basically form this really cool enzyme that is, the number one enzyme in the world, basically. How evolution did that.
Well, it's not just Rubisco. It's almost all of the major enzymes of photosynthesis are partly genetically coded in the chloroplast where they ultimately end up, and partly in the nucleus. And this is how I actually got into plant science, was trying to understand the movement of genes, because the progenitors of plants were simple organisms. They didn't have nuclei and chloroplasts. They had all their genes in one place. You think about a bacterium, all the genes are in one place. So why have genes moved over time, and why didn't they all move? Some of them moved. Because it creates a problem. In your nucleus, you have, maybe 2 copies of each chromosome. If you're a plant, you might have 4 copies, maybe more. But in the chloroplast, you have hundreds of copies, and you have many chloroplasts per cell. So there's a genetic imbalance. You have the potential to make hundreds of times more large subunits than small subunits. So the reason we got into the Rubisco project was to understand how this genetic imbalance was essentially corrected by the plant, and how, so it makes too much large subunit, but why does it let itself do that? And how does it get rid of it? How does it regulate this process? So this was how we got into it. We weren't thinking about turbocharging corn, but once you get into that, it's like a rabbit hole, and you think, wow, well, now we're here. What did we learn? What can we do with that knowledge? And that's sort of how science typically proceeds. You keep going down unexpected places, and nobody's smart enough to figure out where the next, great finding is going to be. So you just follow your nose, and sometimes you end up at a dead end, and sometimes you end up improving corn, I guess.
Yeah, that's really cool. Evolution is neat stuff.
It is, yeah. And we're trying to speed it up here. It's really domestication that we're doing, not evolution. Evolution is if humans would just leave the planet alone, let's see what happens. And then domestication is saying we need to grow more corn on less land. How can we do it? What can we learn from evolution, but speed up in the laboratory, essentially? Did Dave Silverman have another question?
He does indeed. Are there any quantifiable differences among Rubisco found in other grains, such as rice, for example?
Yeah, I mean, this has been a topic of investigation for many decades because Rubisco is so abundant. It's easy to isolate. And so Rubiscos have been compared not just among crop plants, but among thousands of organisms. And what scientists do, and this isn't anything we're doing, but they'll compare Rubiscos from all sorts of different organisms. Oftentimes, the weird organisms are the most interesting. Rice, maybe not so much, but you look at some photosynthetic bacteria or something, and you find Rubiscos with different properties. There's Rubiscos that are really good at discriminating carbon dioxide and oxygen, much better than the enzyme in plants, crop plants. There's some that are very fast. So you can't just take those weird Rubiscos and put them in a plant because they won't assemble. They don't have the right RAF1 and the right RAF2 and all of that. But can scientists take their understanding of the structure differences in them and kind of modify the crop plant Rubiscos to have a slightly different structure? What can we learn from nature that we can apply to plant Rubiscos? There's people on Cornell campus, Maureen Hansen's lab's doing that with the small subunit. There are people doing it with the large subunit. We have a new faculty member down on campus who's studying the crystal structures of Rubiscos and trying to understand those. Her name's Laura Gunn. So I think a lot of exciting things happening in the Rubisco world on the Cornell campus and at Boyce-Thompson Institute. So stay tuned, I guess.
Very nice. We'll do an update in the next year or so, maybe when we have some more stuff, more exciting things. Okay, we're at about time. I'd like to thank you again, Dr. David Stern, for taking the time to chat with us today and educating us about some really cool stuff. And thank you everyone out there who joined us for this new series of virtual chats. I'm going to put a bunch of links in the chat for everyone to see because I'm about to say them, so they'll be there. Please join us for BTI's next Breaking Ground discussion series on Wednesday, January 19. We'll be joined by BTI's Education Outreach Director, Delaney Sickler, who will be talking about BTI's Summer Research Experience Program for undergrads. And we will be joined by two former undergrads who experienced the program, so we will be able to hear their stories directly from them. So that's going to be really good. It's a really cool program. It's unlike a lot of other ones, so please tune in for that. And you can go to btiscience.org for more information and to register. And you can read more about BTI's current research and many other neat stories about BTI Science in our annual report, which you can find online at btiscience.org/annual report. And BTI is an independent nonprofit research institute, and we operate in large part thanks to the generosity of community members like you. And if you'd like to make a gift to support BTI, you can donate online at btiscience.org/give or email our development team at development@btiscience.org. And that's all. Thank you very much for your interest and support of the Boyce-Thompson Institute. And I hope you all have a wonderful day and be well.
Same thoughts. Goodbye. Thank you.
All right. Thank you, David.